you know what the structure of a protein is? Well, the simple answer is there isn't one singular structure. Proteins have multiple structures. They have a primary structure, a secondary structure, a tertiary structure, and a quaternary structure. Now, what I'm going to do in this video is go through each of these distinct structures and dissect them individually to make them succinct, concise, and clear, and make you understand what the hell is going on. Okay, so what's a primary structure? The primary structure is simply the sequence of DNA-based sequences, the sequence of DNA bases that are in a particular polypeptide. So what they do is they're specific to the particular protein that's being coded for. If the DNA base sequence changed, there's a high likelihood that the protein being coded for would change as well. Then this polypeptide coils into either an alpha helix or a beta pleated sheet. This is the secondary structure. Hydrogen bonds begin to form between amino acids, and that, ladies and gentlemen, is your secondary structure. Your tertiary structure is when more hydrogen bonds form between amino acids, but disulfide bridges form whenever two molecules of the amino acid cysteine bond together. You've also got ionic bonds, and ladies and gentlemen, that is your tertiary structure. You know what's interesting about the tertiary structure, though? It's fundamental in determining the shape of the active site of enzymes. Therefore, the tertiary structure has to be very specific. If it's not specific, then the substrate won't be able to bind to the enzyme, and an enzyme substrate complex won't be able to form. If, let's say, I don't know, you were stupid and you heated an enzyme to 100 degrees Celsius, well, what would happen is you damage the ionic bonds, you damage the hydrogen bonds, you damage the disulfide bridges, so the tertiary structure of the active site of the enzyme would no longer be complementary to the shape of the substrate. Hence, that would mean that enzyme substrate complexes wouldn't form. Hence, that would mean that activation energy would not reduce. There we go. I've just gone through an entire 4-marker with you in the span of 10 seconds. Okay, what's the last structure? It's called the quaternary structure. This is when you have more than one polypeptide chain. For example, not all proteins have this, but let's say hemoglobin. Hemoglobin is a particular protein that does have this. So that's it, ladies and gentlemen. That is all the structures of proteins that you need to know: primary, secondary, tertiary, and quaternary. And I hope you enjoyed this video, and thank you for watching.